Thank you, and welcome you guys coming to my talk. And like uh, uh, like you can see, my topic is about metabolic object and trade-offs. And the QR code there is for you to access to our publication that is related to this talk. And like we have mentioned, that we are going to focus on metabolic objective and trade-off, but what are them exactly? And for example, we can see a fish definitely has a fins that is a phenotype that's trying to optimize their objective for swimming. And likewise, we have cats which has 4 legs to optimize their behavior for running. And is there any animal that can optimize the swimming and running at the same time? It does not. I should have used ChatGPT to generate it, but what I can come up with is a Pokemon. Like we can see, like a Pokemon has fins and legs that can run as fast as a cat and swimming as fast as fish. However, it's not real, right? Because the two objectives definitely compete with each other. But there's definitely the animal in reality that's called axolotl that has two different type of phenotype. However, they are competing with each other. So if we take a look at the objective in phenotypical space, let's say swimming and running are 100 out of 100 for fish and cat respectively. They are fully optimizing only one objective, and we also call them archetypes. However, the objective of an axolotl is hypothetically proportional to the two objectives. So let's say they only optimize 80% of the swimming and 70% of the running. And this idea has been well studied in evolutionary biology and ecology. But how about cellular metabolism? Is there any trade-off between metabolic functions? Oh, I forget to say, this kind of competing behavior is called trade-off. And especially for mammalian cells, it's kind of rare to discuss the trade-off or objective inside the cellular functions. And in fact, in the field of metabolic modeling, objective is an important variable to model the reaction fluxes. And we always assume that cell growth is the most important behavior, most important goal to achieve, because lives tend to generate more offsprings and spread their DNA. And biomass objective is most commonly used objective for us to model the reaction fluxes, because biomass would be the surrogate of cell growth. And biomass objective includes all the metabolites necessary for cell to double. And in other words, optimizing biomass objective is equivalent to optimizing cell growth. However, yeah, it's true that this assumption is widely accepted to model mechanistic state in microbiome. But there are a lot of issues that makes the use of biomass objective questionable. First of all, the idea is definitely questionable, especially for stem cell, because we all know stem cell, they do not divide in adult. They are mostly quiescent. Otherwise, it's gonna be a cancer or something uncontrollable. And the second issue is that there are actually 40-ish metabolites inside the biomass objective. And like I'm showing here, I won't go into detail, but mathematically, biomass objective is the linear combination of those 50-ish metabolites that I'm showing on the bottom of the slide. For example, we can see the ATP has 20.7 for the coefficient and 0.53 for glycine, and so on until they finish the formula with the 40-ish metabolites. So they definitely have different coefficient. However, is that true? We can keep the coefficient the same for all type of different cells? It's definitely weird. And let's say if we only pay attention to glycine and ATP here, bacterial cell would choose probably higher glycine to promote their growth, and stem cell would choose more ATP to support their maintenance. So obviously, in the phenotypical space, they would compete with each other, then choose different weight or different coefficient for different metabolites. And the same idea also showed up among like metabolic functions. For example, cell growth can compete with energy production and redox balance. It's kind of intuitive because high growth can result in higher like oxidative stress. Then the cell has to produce or promote more like redox balance function to protect the cell. And in other words, like if the cell promote more like redox function, it would compete with like the biomass objective. Then you would like cancel out the like cell growth. And to really find out the metabolic objective for different conditions and different cell types, like I was mentioning, and considering all the issues I was mentioning before, we hypothesized that we can rely on this like 52 potential components. They are all metabolized as component to infer any type of metabolic objectives to represent phenotypes, no matter it is redox balance, cell growth, cell migration, or cell death. And just like biomass objective, we will have an infer metabolic objective, which is a linear combination of these metabolites, like I'm showing on the bottom. But their coefficient can reduce to be 0, then remove the certain metabolites. So it won't be like a biomass objective that fixes the coefficient for each metabolite. And to find out the equation, like we said, for the infer metabolic objective, we developed a tool called SCUDI. And SCUDI takes cell-specific omics data as an input. Then we can use the cell-specific like omics data as a like constraint to model the reaction fluxes. After we simulate the fluxes, we can use like a machine learning model to extract the metabolic feature from the reaction fluxes and output the metabolic objective as a outcome. And in fact, the cell-specific objective flux prediction is one of the 2 parts of SCUDI, and we call it constraint models. And a constraint model reflects the most possible flux route regulated by metabolic genes. And those metabolic genes are like cell-specific. And to model the metabolic flux, we have a knowledge-based like model called RECON1. I guess everyone is really familiar with that. And it's constructed based on the known reactions, enzyme, and the metabolic genes from human cells. And like I say, we want to get like cell-specific fluxes. Therefore, we constrain RECON1 by the gene expression data. And for instance, we have a simplified RECON1 on the right-hand side. And suppose we have like a upregulated genes, which is showing in red. They can control the reactions, and the flux can be blocked by the blue genes. Then the flux will be rerouted, and like I'm showing in green, it will be the final like flux route. And the RECON1 model is similar to Google Map, which was built by previously collected information like roads and cities. And we can always find the best route by setting up our destination to a location. Like there's a destination like a point from like hell to paradise in Michigan. And the basic Google Map suggests the best route to go to the paradise. And we found like the objective in Google Map is usually the shortest distance and the shortest traveling time. And the genes actually here like the obstacle on the road that block you to access a certain city or the certain route. So the gene kind of like redirect the flux to guide you to find the best like metabolic flux. And after we got the cell-specific fluxes, as known as the constraint model, we are almost ready to infer the objectives. And in addition to the constraint model, we need the unconstrained model that is actually the ideal optimal solutions. And for example, the first model only optimizes ADP and get optimal route for metabolic fluxes. Then we can do the same thing for optimizing glycine. It's a map to show like as much production as like glycine as possible. And we can keep doing the same thing for the 55 different metabolites. Then we will get like the like a matrix that are showing actually the best like different flux prediction for different objective. So using like Google Map as a metaphor, there are like 52 different goals like shortest path, cheapest fee, or the most beautiful landscape along the way. They are just representing different type of objective for the unconstrained model. And because we don't know the real objective used by different cell, different conditions, So we treated the unconstrained model that optimized different objectives to be the variable to fit the constrained model predicted by the different transcriptomics data. Then we can use like a meta-linear regression model that estimates the coefficient for each like candidate objectives. And eventually the model will output a metabolic objective for each condition. And to test our model, we firstly use like quiescent versus like proliferative cell, because it's more like intuitive, because quiescent cell, we know like it obviously showing like lower growth rate, and proliferative cell instead divide faster. And this is an important like cellular behavior also for like cancer or stem cell. And like I say, we know that a biomass objective represent the cell growth phenotype. So we can compare the inferred objective coefficient to the biomass objective by Euclidean distance. And each value here, and each value here is like, if the distance is closer, it means that it is closer to like the cell growth phenotype. And as a result, it's really cool that we did see cells in proliferative states had like objective that show like significantly shorter distance that is closer to growth phenotype. It looks like it looks really making sense, but it's like we are really improving that like our metabolic objective can like validate that and to identify different phenotypes, especially for like cell growth. Then we introduce the single cell cycle data to verify if we can extract metabolic feature by metabolic objectives. And if we directly compare the coefficient from the metabolic objective in like G1, S, and G2 phases, we can find that we can identify like significant metabolites in the three cell cycle phases. And in G1, we can see glycine, tyrosine, NMN, which are related to growth boosting metabolites, and they are all identified significant here. And it aligns with the consensus that G1 produce more metabolites to support higher growth. And G2 alternatively has significantly higher coefficient of lipids and proline, which are all related to structure and fatty acid. And likewise, we know that G2 is in charge of like synthesizing cell membrane and structure like metabolites or biomaterial. And lastly, we know that DNA synthesis and the replication are active in S phase. And we also see that the CTP as a nucleotide and the glutamine are all like significant in the S phase. And the association between this phase and nucleotide synthesis is like recapitulated the behavior. And we were super excited to the result. And so far, like Scooty can infer the metabolic objective to represent any phenotype and extract the metabolic features. And in the second part, we will focus on embryogenesis, especially for the early part of the embryogenesis. And we wonder what the objective is and how the objective can lead to the underlying developmental processes. And we focus on two transitions. One is zygote to 2-cell, called 1C, 2C, and another part is like 2C to 2-cell to blastocyst, we call 2CBC. And the reason why we focus on the two transitions is that gene expression is mostly quiescent in zygote, especially for no transcription. And it's mostly rely on like material, like maternal material and pyruvate metabolism until the 2C stage. And 2-cell is the same, it process, there's a process called like zygotic gene activation. They initiate like transcription after like the 2-cell stage. And after 2-cell stage, we can see that there's like a huge change, like especially for differentiation and divided into like inner cell and the outer cell like space, like cell type. And we obtain the single cell data for the early embryogenesis, and we use them to infer cell-specific metabolic objectives. And relying on UMAP, the metabolic objective can be projected onto the two-dimensional space. And as we can see, they are separating into two parts roughly. And if we take a closer look at the top right corner, and we do the enrichment analysis, we found that the top right corner cluster is enriched in pluripotency. And the bottom part is enriched in like cell growth or proliferation. So we can understand this result by this illustration. There are two metabolic objectives as long as like metabolic trait, they form like trade-off to guide the transition during the embryogenesis. And in the end, we can use the figure again to summarize the trade-off with the illustration. And apparently, the trade-off between the two phenotypes were like revealed like by choice of objective. So like by the competition, they optimize a certain like phenotype to reach their goal. And like the take-home message is like we can use Scooty, which combine the mechanistic and machine learning model to unveil the cell-specific objective. They can represent any phenotype and also extract metabolic objective and metabolic features. And we apply the Scooty to find out the objective that suggests a trade-off and switch of the metabolic function from development to growth. And in the end, it's actually what we are working on right now. We use like trade-off analysis and objective to pave a new way for cell type separation, hypothesis fighting, and disease and drug like discovery. And of course, the last take-home message is that it's also a trade-off to choose my talk. And thank you all to prioritize your time to sit in here.
Lisa, I see already.
Fantastic talk. I have a question about the implementation of the single cell analysis. So dropouts are a really big problem with single cell transcriptomics. Are you filtering out cells that have dropouts of the metabolic enzyme expression, or are you imputing the values when they're not present?
I impute them, yeah. But I also remove like the cell they are now like probably expressing the metabolic genes because for our model, like especially using like metabolic modeling, we mostly only focus on like metabolic genes. So if they don't like express metabolic genes, I would just like get rid of them. And yeah.
Is there the danger of biasing your sort of objective phenotype because of that?
Fortunately, for this data set, I remember I only like removed like 3 cells in total. So this data set is pretty clean, I guess, because it's a process data. So I guess maybe they have been processed, and so there's not a lot of issue. And but we also try them out. Like we are not only try out this data set, we also try out in another single cell data set, and it works like similar. So I guess that's not a big issue for embryo. However, like you say, I also try this method in other like cell type, like EMT or something, and it's not working that well. But in embryo, I don't know why it always like working really good, because I found like the trade-off is pretty clean. Maybe there's a reason because like the resource in embryo is kind of limited, so that's why they kind of prioritize a certain like certain genes and certain like motif or something like that. Yeah.
Thank you very much.
Yeah.
Maybe I also have a question. When you say that when you're applying the tool, you are able to retrieve something that you would expect, for example, in the G1 phase or in the S phase or the G2 phase. Do you see something that you would not expect, but that could be interesting to test it as a new prediction from your tool to the experimentation, for example?
Actually, I have a cool like finding. I mean, I did because I have a bunch of data. I didn't show them out, and it's like I use like the UMAP again to kind of like separate the like different phases, and I found it's kind of like cool that we can find like a trajectory of like the 3 different phases. However, they are not following the like the order like G1, G2, G1S, G2. It's like G1, G2S. Yeah, but I didn't like because it's like our like testing data. We mainly focus on embryogenesis, but for this one we found like we can still find out like the change of like the objective or the trade-off like step by step, like in like during the like cell cycle phases. Yeah.
Thank you very much. Further questions? If not, let's thank again our speaker.
Oh, sorry.
It's a quick one. Could I just ask you,
Yeah, sure.
the transcriptomic constraints?
Yes.
What method did you use to integrate Was it a valve-based method or a switch-based?
It's like I use like a couple different like methods, but the method I mainly use is like it's called linear IMAP. It's like I constrain the reaction associated with the metabolic genes, and if like it's upregulated, I would just like like turn on the reaction, and if it's like downregulated, I would just like close the gene like the reaction, so it will just block the flux. Yeah, so it's like the simple way. But I did try like many different like methods to test if they affect the result. But speaking of the objective or the trade-off, they are pretty consistent. The pattern is similar. However, we did find out like different like metabolites or different coefficient inside if we switch the method. Yeah.